Ladies and gentlemen, today we are going to talk about our project. Here is the outline of our presentation. The most annoying part during the health check is that we cannot eat anything except water. After doing some research, we discovered that the reason is about some psychostimulants and depressants, which can affect our endocrine. Understanding these effects can explain why testing is required before health checks and how these substances influence our body's normal functions. So, in order to find out how psychostimulants and depressants affect us, we come up with the research. With this in mind, we focus on dyspnea as our subject. First, their small size and accessibility simplify preparation. Second, their transparent bodies and beautiful heartbeats facilitate real-time observation. Finally, their cardiac response to chemicals mirrors the pharmacological reactions in higher levels. Consequently, dyspnea was chosen as the ideal model to investigate the physiological effects of these substances. Our study has three main goals. First, to investigate the effects of different drugs on the heart rate of dyspnea. Second, to examine the influence of various environmental factors on the heart rate of dyspnea. Third, to investigate the antagonistic effects and interactions between chemical substances and environmental factors on the heart rate of dyspnea.
This is our literature review. This study looks at how alcohol and caffeine affect dyspnea heart rate. Alcohol slows heart rate, also at a higher concentration. Caffeine speeds it up, but the effect depends on concentration and environment. When both are present, they may cause an antagonistic effect. Dyspnea may also have a homeostatic response to prevent heart rate from rising too much. Research on their combined effect is still limited, so this study investigates their interaction.
And then we'll introduce the agonist and antagonist. Agonist is a substance triggers a physiological response in the receptor. You can see this picture. With agonist combined with the receptor, you will have action. On the other hand, the antagonist is a substance that blocks the action of an agonist. It's also combined with the receptor. However, it don't have any action.
These are the equipment we use, including dyspnea, smartphone, video editing softwares. We also use 95% alcohol and pure caffeine powder. Finally, our observation tools including compound microscope, concavity slide, cover slip, petri dish, and digital probe thermometer.
And then is our experiment design. First, we use room temperature dyspnea as a control group. And then we design 6 treatment groups, including ice water, caffeine, and alcohol. And then we mix both two of them and become solution like ice alcohol solution, ice caffeine solution, and caffeine mixed alcohol solution. And this is our step of experiments. First, we prepare the resolution we need, and second, we soak the dyspnea into the solutions and wait for 10 minutes. After 10 minutes, we take the dyspnea under the microscope and make an observation, and as that, we count the heartbeat. We use a smartphone to take 10-second videos and count the heartbeats, and after we collect the data, we use Excel to make charts.
Next is our result. First is the ice water group. Blood vessels contract in the low temperature environment, which makes it harder for blood to flow. So the heart needs to beat faster to keep the blood circulation and maintain body temperature. As a result, the heart rate is increased. Next is the ethanol group. Ethanol slows down the central nervous system and strengthens the effect of GABA, which is an inhibitory neurotransmitter. When GABA binds to the receptor, it opens ion channels. This allows negatively charged chloride ions to enter the cell and positively charged potassium ion to leave the cell. This process is called hyperpolarization. Hyperpolarization makes neurons less active and reducing firing rate. As a result, the heart rate is decreased.
and then it's a caffeine group. Caffeine inhibits an enzyme called phosphodiesterase (PDE), and which normally breaks down the cyclic adenosine monophosphate (CAMP). When PDE is inhibited, the level of CAMP increases, so which triggers a protein kinase A (PKA). PKA regulates ion channels through phosphorylations and increase the ions movements such as calcium ions. So that's increase the heartbeat of cardiac muscle cells, so the heartbeat increase.
Next is the ethanol group. First is the caffeine pro-ethanol group. Caffeine directly speeds up the firing rate of cyanoethanol. On the other hand, ethanol affects the heart rate more indirectly by slowing down the central nervous system, and the effect is slower and more widespread. So when both are present, caffeine has a stronger effect, so the heart rate is increased. Finally, when ethanol or caffeine combine with ice water, low temperatures slows down the rate of variations. So the change in heart rate, whether increase or decrease, are smaller compared to the group with only ethanol or only caffeine. Next is our future direction. First, we want to study whether long-term use can change the resting heart rate. Second, drinking energy drinks or alcohol at the same time may either cancel each other out or have a combined effect. Third, increasing the sample size will make our results more reliable and more general. These are our references.
This is the end of our presentation. Thank you.